Today I'm going to do a monohybrid cross. I have a limited amount of space, so I'm going to try and use the edges a little bit to help explain. I would normally write this below one line above the other, but Mr. English's golden rules are: write down what you know, work neatly, and answer the question. Those are the three rules that you must try and follow. Right, now in this particular case I'm going to say tall is dominant and short is recessive. Now, that being the case, we must assign letters. I'm going to come across over here. The capital T is for tall and the lowercase t is for short. What I'm going to do is I'm going to say, let's take a purebred tall plant. This is what Mendel did. We're going to cross it with a purebred short plant. Now when we say purebred, we mean that there's no other genes being carried here. There's only dominant genes. So those are two big T's and two small t's. Both of these are purebreds. Let's cross them together and see what we get. Over here, and this is very important how you lay it out, so please watch carefully. P1 are the parents. So we put the parents into boxes like this, and we're going to cross it with another parent like that. So we're going to cross a purebred tall with a purebred short. Then what happens to get the gametes, we need this process called meiosis. So it's okay to write meiosis there, and then over here we've got to write the word gametes. So I'm going to move across a little bit just to give me some space. Now the gametes are always represented in a circle. So I'm going to draw them relatively large, and for each individual there must be two gametes. So Mendel's law says that one of those two, one of this pair of alleles, must be in each of the gametes. So big T in there and big T in there. And exactly the same goes for this purebred short plant. It's very important to write both options, even though you're replicating yourself. I prefer that if you do. Now we're going to draw a Punnett diagram, and that's called the first filial generation. And the first filial generation I want to represent in a punnet. So a punnet is like a tray, and in this tray we are going to put the cross between the two. So these gametes over here go into that block, this one into that one, and big T into there, and big T into there. So effectively we're going to cross that with that. Let's do it slowly. Big T, small t, big T, small t, big T, small t, big T, small t. Great. So now we've finished the F1 generation, but we haven't really finished because we need to write down the genotype. Now the genotype is expressed as a ratio of all the possibilities in this box. And you'll notice that there's only one possibility, so for the whole F1 generation, they all have the genotype of big T, small t. Now the phenotype, you write this out, and again it's expressed as a ratio. But if you look carefully, there's only big T, small t, and that means each one of those will be tall, because the tall gene dominates over the recessive gene, the short recessive gene. And so in the phenotype you've got to express what they look like. And we will write here, they're all going to be tall. Right, the next step is to cross the F1 generation with itself. Now it's not always going to be the case that you have to do this, but they may in the question tell you or ask you to cross the F1 generation with itself so we can get the F2 generation. So now what we write is P2. So these are the second parents, and I'm just going to write them out and then give you a little explanation. Now from the F1 generation, we had big T and small t plants, but we only had big T, small t genotype plants, and they were all tall. So now we take each one of those, any two of those, and we're going to cross them, and we use the same rules as above. Over here we write out the word gametes, and we draw circles for the gametes. So this one we're going to write big T and the other one small t. Now you see how that differed to the P1 generation, their gametes. Here we've got a tall gene and here we've got a short gene. They're opposite traits, they're opposite alleles of the same gene. It had to come from that particular parent. And this one is identical. Right, now we're going to write the F2 generation. And again, we're going to draw Punnett diagram. Always try and get these fairly big. These gametes are going to go into the top row, big T, small t, and these gametes are going to go into the vertical row, big T, small t. And now we can cross them, big T and big T, big T and small t, big T and small t, and small t and small t. And now we see a different combination of genes compared to the F1 generation. Let's write out the genotype of the F2 generation. This is important, you must do it every single time. Remember, working neatly is important. Genotype and the genotype of this. Now we have to write them again as a ratio and write all the possibilities. I'm going to start there. So it'll be big T is to big T, well, sorry, big T is 2. Now there's 2 of these, so we're going to write 2 big T, small t. Now you don't write 1 big T, like in the maths you don't write 1x, you just write x, but in 2x you write 2x. So it's the same principle. And then lastly, we have only one of these, small t, small t. So that's going to appear as a tall plant. These are both going to appear as a tall plant, and that's going to appear as a short plant. Let's write out the phenotype. Right, so there's tall, and then another 2 talls and 1 short. So it's 3 tall to 1 short. I'm a little in doubt whether to write 1 there, but I think it's be fair enough to write 1 short. Now, that was the unusual outcome that Mendel got. When he inbred the F2 generation, so the F1 generation, creating the P2 generation, he got this amazing outcome of 3 talls to 1 short, always in that ratio. And this is how we'd express a full genetics problem, remembering if they ask you to inbreed the F1 generation from that previous slide or that previous video.